Hello and welcome. Today we're going to embark on an exciting journey into the world of quantum dots labeling proteins for detection. I'm from CD Bioparticles, and let's get started exploring this cutting-edge technology. To begin with, let's clarify what quantum dots are. Quantum dots are semiconductor nanocrystals, incredibly tiny, typically sized between 2 to 10 nanometers. This minuscule size gives them quantum confinement effects, which result in size-dependent optical and electronic characteristics. These unique properties set them apart in the scientific world. Now, why is protein labeling so important? Protein labeling is a cornerstone in biological and biomedical research. It allows scientists to detect proteins within cells and tissues, track their movements, and study them in detail. By doing so, we can gain valuable insights into biological processes, understand disease mechanisms better, and even develop more effective drugs. Finally, let's talk about the role of quantum dots in protein labeling. Quantum dots have emerged as powerful tools in this area. Compared to traditional fluorophores, they have superior fluorescence properties. They are highly bright, photostable, and their emission wavelengths can be tuned. This makes them ideal for protein labeling, opening up new possibilities in research. Let's take a closer look at the properties of quantum dots. First, their optical properties are truly remarkable. The fluorescence emission of quantum dots is size-tunable. Smaller quantum dots emit light at shorter wavelengths, in the blue-green range, while larger ones emit at longer wavelengths like red near-infrared. Their narrow emission spectra and high quantum yields mean that the fluorescent signals they produce are intense and distinct, making them easy to detect. Quantum dots also have unique electrical properties. They possess high electron mobility and can act as electron donors or acceptors. These electrical characteristics are exploited in applications such as fluorescence resonance energy transfer or FRET-based assays. When we compare quantum dots with traditional fluorophores, the advantages of quantum dots become even more apparent. Traditional fluorophores, like organic dyes, suffer from photoleaching, which means their fluorescence fades over time. They also have broad emission spectra. In contrast, quantum dots are more photostable, enabling long-term imaging. Their narrow emission spectra allow for multiplex detection, where multiple proteins can be detected simultaneously. Now that we understand the properties of quantum dots, let's move on to the first step in the protein labeling process: surface functionalization of quantum dots. Quantum dots synthesized in organic solvents usually have hydrophobic surfaces. This is a problem because biological environments are aqueous. So, these hydrophobic quantum dots are initially incompatible with the biological samples we want to study. To solve this, we use specific functionalization methods. Molecules like thiols or carboxylic acids are attached to the quantum dot surfaces. Thiol-coated quantum dots can form strong bonds with metal atoms on the quantum dot surface. On the other hand, carboxylic acid functionalized quantum dots are perfect for further conjugation reactions with proteins. The impact of this functionalization process is significant. It improves the solubility of quantum dots in water, making them suitable for interacting with biological molecules. This ensures that quantum dots can be effectively conjugated with proteins later on in the process. After functionalizing the quantum dots, we need to modify the proteins. Why is this necessary? Well, native proteins often don't have the appropriate reactive groups for direct conjugation with functionalized quantum dots. So modification is key. Common methods for protein modification involve introducing amine or thiol groups onto the protein surface. This can be achieved through chemical reactions with specific reagents. For example, to introduce an amine group, we can target lysine residues in the protein, and for thiol addition, we can target cysteine residues. But it's crucial to preserve the protein's function during this modification process. We don't want to denature the protein or significantly affect its biological activity. That's why we use mild reaction conditions to ensure the protein remains functional. With the quantum dots functionalized and the proteins modified, it's time for the conjugation reaction. There are different conjugation chemistries available. Amine reactive groups, such as N-hydroxysuccinimide esters, can react with the amine, modified proteins and quantum dots, forming stable amide bonds. Thiol reactive groups like malimides are also useful. They can conjugate thiol, modified proteins and quantum dots. The conjugation reaction needs to be carried out under specific conditions. This includes the right pH, temperature, and reaction time. For example, the reaction between amine, modified quantum dots and proteins often works best at a slightly basic pH, around 7 to 9. This optimal pH promotes efficient coupling between the two. Ensuring specificity and stability in the conjugation is of utmost importance. The chosen conjugation chemistry should create a specific and stable linkage between quantum dots and proteins. This stability is crucial because it maintains the integrity of the labeled proteins during detection and analysis. After the conjugation reaction, our mixture contains unreacted quantum dots, proteins, and other reagents. This is where purification comes in. The purpose of purification is to isolate the quantum dot, labeled proteins, and remove all these contaminants. There are several purification methods. Size. Exclusion chromatography separates molecules based on their size. Dialysis uses a semi-permeable membrane to get rid of small molecules and unbound components. Centrifugation can also be used to separate larger conjugated particles from smaller, unreacted ones. Purification is not just an extra step, it's a critical part of quality control. High-purity labeled proteins are essential for obtaining accurate and reliable detection results. Without proper purification, our results could be inaccurate and misleading. Once we've purified the labeled proteins, we need to characterize them. Spectroscopic analyses are very useful here. Absorption spectroscopy can confirm the presence of quantum dots by detecting their characteristic absorption peaks. Fluorescence spectroscopy, on the other hand, measures the fluorescence intensity, emission wavelength, and quantum yield of the labeled proteins. This helps us verify that the quantum dots are still functional after conjugation. There are also other characterization techniques. Transmission electron microscopy, or TEM, allows us to visualize the size and morphology of the quantum dot protein conjugates. Dynamic light scattering, DLS, provides information about the size distribution of the conjugates in solution. By analyzing the results from these characterization techniques, researchers can assess the success of the conjugation process. They can determine the degree of conjugation and check the integrity of the labeled proteins. Now that we have characterized the labeled proteins, we can use them in various assays. Immunofluorescence is one common method. It allows us to visualize the labeled proteins within cells or tissues using a fluorescence microscope. Western blotting is another powerful technique. It's used to detect specific proteins in a complex mixture. First, proteins are separated by size, and then we use quantum dot labeled antibodies to probe for the target proteins. ELISA, or enzyme, linked immunosorbent assay, can quantify the amount of target protein in a sample. Quantum dot labeled proteins offer several advantages in these assays. Their bright fluorescence gives high sensitivity, making it easier to detect even small amounts of proteins. Their photostability enables long-term and repeated measurements. And because different colored quantum dots can be used, multiplexing is possible. We can detect multiple proteins simultaneously. However, there are also limitations and challenges. One major issue is a potential non-specific binding of quantum dots. This can lead to false positive results. Additionally, the cost of quantum dots and the complexity of the labeling process can be limitations in some applications. Let's now explore the applications of quantum dot labeled proteins, starting with cell imaging and tracking. Quantum dot labeled proteins are incredibly useful for visualizing protein localization within cells. For example, if we label a membrane-bound protein with quantum dots, we can precisely determine its distribution on the cell membrane. The photostability of quantum dots is a game changer when it comes to tracking protein dynamics. It allows us to track protein movement within live cells over long periods. This provides valuable insights into processes like protein trafficking, which is crucial for understanding how cells function and respond to stimuli. Using confocal microscopy or live cell imaging systems, researchers can even observe the dynamic behavior of proteins in real time. They can capture events such as protein-protein interactions and conformational changes, which are key to understanding cellular processes at a molecular level. Another exciting application is multiplex protein detection. The principle behind multiplexing is quite simple. Different size quantum dots can be conjugated to antibodies that target different proteins. Each quantum dot emits light at a distinct wavelength. This enables us to simultaneously detect multiple proteins in a single sample. Why is this important? In a cell, multiple proteins are involved in complex signaling pathways. Multiplex protein detection using quantum dots allows researchers to analyze the expression and activity of these multiple proteins at the same time. This provides a more comprehensive understanding of these biological processes. For example, in cancer research, multiplex detection can help identify unique protein signatures associated with different cancer types. This could potentially lead to more accurate diagnosis and personalized treatment for patients. Quantum dot labeled proteins also play a significant role in biosensing and diagnostics. They can be incorporated into biosensing platforms, such as immunosensors. These sensors work by detecting the binding of a target biomolecule, like a protein biomarker, to the quantum dot labeled antibody. When this binding occurs, it results in a change in the fluorescence signal. In clinical diagnostics, quantum dot-based biosensors can be used to detect disease-specific biomarkers in patient samples, such as blood or urine. For instance, detecting elevated levels of certain proteins can be an indication of diseases like cancer or Alzheimer's disease. Quantum dot-labeled proteins are not just useful in the medical field, they can also be used for environmental monitoring. By binding to specific molecules in water or soil samples, they can provide a sensitive and rapid method for detecting pollutants or contaminants. Next, let's look at how quantum dot-labeled proteins are used in fluorescence resonance energy transfer, or FRET. The FRET principle is based on the transfer of energy between a donor fluorophore, in this case, a quantum dot, and an acceptor fluorophore when they are in close proximity. When the donor is excited, it transfers energy to the acceptor, which causes a change in fluorescence properties. This principle is applied to study protein-protein interactions. By labeling two interacting proteins with a quantum dot as the donor and an appropriate acceptor fluorophore, researchers can use FRET to study how these proteins interact. Changes in FRET efficiency can indicate conformational changes or binding events between the proteins. FRET can also be used to monitor the assembly of complex molecular structures, such as protein complexes or nucleic acid protein complexes. It provides insights into the step-by-step formation and stability of these assemblies, which is crucial for understanding molecular biology. Moving on, quantum dot-labeled proteins have promising applications in drug delivery and theranostics. Functionalized quantum dots can be conjugated with therapeutic agents and targeted to specific proteins or cellular compartments. For example, in cancer treatment, quantum dots can be designed to bind to receptors overexpressed on cancer cells, delivering drugs directly to the tumor site. In theranostics, quantum dots serve a dual purpose. They can act as both imaging agents and carriers for therapeutic payloads. This allows for real-time monitoring of drug delivery and treatment response. For instance, in cancer treatment, quantum dot-based theranostics can help doctors visualize the tumor and track the effectiveness of chemotherapy. However, although these applications are promising, there are challenges. Ensuring the safety and efficient delivery of quantum dot drug conjugates is a major concern. Future research aims to overcome these challenges and develop more effective quantum dot-based theranostic strategies. Now, let's discuss how quantum dot-labeled proteins are used in flow cytometry and high-throughput screening. Flow cytometry is a technique that analyzes the physical and chemical characteristics of cells as they flow through a laser beam. Quantum dot conjugated antibodies can be used to label specific cell surface proteins. This enables the identification and quantification of different cell populations. In high-throughput screening, flow cytometry is a powerful tool. It can rapidly analyze a large number of cells, providing information about protein expression levels and cellular phenotypes. This is extremely useful in drug screening, where researchers can test the effects of multiple compounds on cell populations. Compared to traditional staining methods, quantum dot conjugated antibodies offer distinct advantages. They have better sensitivity and multiplexing capabilities, allowing for a more comprehensive analysis of cell populations. In vivo imaging is another important application of quantum dot-labeled proteins. In preclinical research, quantum dots are used to image labeled proteins and cells in animal models. In some clinical studies, they are being explored for non-invasive imaging of human patients. This type of imaging can provide real-time information about biological processes. For example, it can monitor angiogenesis, which is the formation of new blood vessels, or immune cell trafficking. However, there are challenges in using quantum dots for in vivo imaging. The biocompatibility and toxicity of quantum dots in living organisms are major concerns. Ensuring that quantum dots do not cause adverse effects while still maintaining their imaging properties is a significant challenge. Let's now shift our focus to how quantum dot-labeled proteins can be used for DNA detection. There are two main methods. One is through fluorescent resonance energy transfer, FRET. In this approach, DNA is coupled to an appropriate fluorophore. The interaction between the quantum dot protein complex and DNA causes changes in quantum dot fluorescence. The other method is basic fluorescence measurements of quantum dots. Here, the binding of DNA to the quantum dot protein complex alters the quantum dot's fluorescence properties. This DNA detection using quantum dot-labeled proteins is very important in fields like genetics and genomics. It can be used to detect specific DNA sequences, such as disease-associated genes. It's also useful for studying DNA-protein interactions. Future research in this area may focus on improving the sensitivity and specificity of these DNA detection methods, as well as developing more efficient ways to couple quantum dots with DNA-binding proteins. Now, let's take a look at some related products. CD Bioparticles offers carboxyl PEG functionalized cadmium selenide zinc sulfide quantum dots, which are designed for protein labeling and DNA detection. These quantum dots have a PEG carboxyl coating, and this coating is highly beneficial for bioconjugation. The PEG coating has several advantages. It improves the water solubility of the quantum dots and reduces nonspecific binding. The carboxyl group on the coating can react with amine groups on protein surfaces, forming stable amide bonds for conjugation. These quantum dots also have great features. They are monodisperse, meaning they don't agglomerate or aggregate. They have a tunable emission range from visible to near-infrared wavelengths. They've been extensively characterized using TEM, ultraviolet visible, and DLS spectroscopy. They also have high fluorescence intensity, good stability, and a long lifetime. In conclusion, quantum dots have truly revolutionized protein labeling for detection. They offer unique properties and a wide range of applications, from cell imaging to DNA detection. The labeling process, which includes steps like surface functionalization, protein modification, and conjugation, is complex but crucial for successful detection. As research continues, we can expect quantum dots to play an even more significant role in biological and biomedical research, diagnostics, and therapeutics. However, we must address challenges related to biocompatibility, toxicity, and cost. So, I encourage further research and collaboration in this field. By working together, we can fully explore the potential of quantum dot-labeled proteins and overcome the existing challenges, leading to new breakthroughs in science and medicine. Thank you.